In the overall process of cellular respiration, foods, including sugars, and as we will be seeing, even fats, and even proteins, can be broken apart. Now, there are these covalent bonds that hold the carbon and hydrogen atoms together, and oxygen atoms together in these sugar molecules and fat molecules and protein molecules. When these covalent bonds are broken, meaning we're breaking the molecule apart into smaller and smaller pieces, that's what releases the energy. So the energy is contained within these organic molecules by virtue of these covalent bonds that hold these atoms together. Now, we are breaking these molecules apart to release the energy, and that energy that's released is used to make ATP. ATP, we said, is the gasoline that powers cells. And so we have to take the energy that's in sugars and convert them into ATP molecules. And I used as an analogy last time that it's a little bit like a change machine where you convert a $5 bill, a $10 bill, a $100 bill into quarters. And quarters are actually like the ATP molecules that are actually used as sources of energy. So as I point out in my video, if I were to ask you on a test, which I'm very likely to do, which contains more energy, one sugar molecule or one ATP molecule, sugar contains far more. There's enough energy in one glucose molecule to produce 38 ATP. So obviously it contains a lot more energy. But just like washing machines and dryers seem to, in a coin-operated laundromat, only seem to run on quarters, the biochemical processes in living things run on quarters, called ATPs. So we have to turn $10 bills into quarters. Now, of course, whenever you need more ATP to power, let's say, your muscles as you start to exercise and start to run, so that's going to cause this whole process to speed up. It starts speeding up as you start to exercise faster and faster. So as I pointed out on my video that's posted on YouTube with 110,000 hits, is that as you start to exercise, you start breaking down sugars at a faster rate to release more energy to make ATP at a faster rate. You also start inhaling oxygen at a faster rate. You also start exhaling carbon dioxide out of your mouth at a faster rate, because that's a waste product form. The sugars are broken apart into carbon dioxide and then exhaled out. And you also get hot, because, in fact, over half the energy that's released, that's in that sugar molecule, is released in the form of heat. And that, we said, was predicted by the second law of thermodynamics that we talked about on page F4. That whenever you convert one form of energy to another form of energy, some of that energy goes off as heat. In fact, the majority of the energy. So that really is explaining why, when you exercise, you burn food, right? You burn up calories of energy in the form of sugars and fats. You inhale more oxygen. You exhale more CO2, and you get hot. So that's why it's kind of a fundamental statement about energetics, or energy use, energy production, and the use of energy in all living things, including us. Now, we also talked about last time the regulation of our blood sugar level by two protein hormones, both produced by our pancreas, insulin and glucagon. These are the yin and yang of these two hormones. One raises the blood sugar level, the other lowers the blood sugar level. Which one raises our blood sugar level? Glucagon. Which one lowers it? Insulin. Do you think I'm going to ask you that on a test? 100% probability. Do you think I'm going to ask you, in terms of a multiple choice question, what glycogenolysis is, or glycogenesis? Right. So I'm just trying to, in case there's any doubt in your mind, do you need to know these words that we've talked about? Make sure there's no doubt in your mind. We will ask you what those words mean. Now, we then started to talk about this whole cellular respiration process on page H10. And on H10, our first thought is, that's a scary looking diagram. Actually, if you look at your textbook, they actually have scarier pictures than this. I'm trying to keep this simple. But we raised the question last class meeting on Monday, why don't we, if we're going to break apart the sugar into carbon dioxide and release the energy that's in the sugar to make ATP, why don't we just do it in one single step? Just release all the energy break apart that glucose into 6 carbon dioxides in one single reaction. Why doesn't the cell do one single reaction? Why does it have to do all of this? You'll notice we learned last time, first the glucose is split in half, turns into 2 pyruvate sugars. Today we're going to learn that they're going to be broken in half, and we're going to keep breaking this thing apart step by step by step until eventually it's broken apart into 6 individual carbon oxides. Why do we use all these little intermediate steps? Why not just one single step? So we don't blow up. Because otherwise it would be like igniting gasoline inside of a cell and watching it blow up. All right? Because that would be an explosive release of energy, and we need a controlled release of energy. So inside of a car engine, inside the chambers of that engine, the cylinders, there you literally have explosions occurring that are moving the pistons up and down, but we're not made out of metallic parts and iron chassis and so on. So it has to be step by step by step. We divided cellular respiration into 2 phases, anaerobic reactions that occur in the cytoplasm. They do not require oxygen. Oxygen can be present or not present, doesn't really matter. And then that's followed by aerobic respiration reactions that require oxygen. These occur in the mitochondria. So in summarizing what we learned last time, we said that the main thing that's occurring in the cytoplasm in the anaerobic respiration reactions is something called glycolysis. Lysis means to break apart. Glyco means glucose, to break apart glucose. And all that's happening is glucose is being split in half. It goes from a 6-carbon glucose molecule, C6H12O6, to 2 separate 3-carbon sugars called pyruvate sugars, or pyruvic acid. Now, that's basically all that happens in the cytoplasm. Now, that does release some energy by breaking the sugar in half, and that's enough energy to have a net gain of 2 ATPs. And we talked about gross income and net income last time. We grossed 4 ATPs, but we netted 2 when we take into account what it took to initiate the process. In addition, some NADs, which are the coenzyme that contain the B vitamin niacin, have each picked up a pair of what I call hot potato hydrogen atoms off that sugar molecule. Now, we know that the whole reason why we breathe oxygen is because we got to do something with these hydrogens that are being broken off the sugar molecule. We can't just release the hydrogens either into the cell or in the area surrounding the cell, because that would increase the acidity. The definition of acidity, or pH, is hydrogen atoms. So basically we breathe oxygen, so does your dog, so does your cat, so do all living things in general, and they attach those hydrogens, 2 hydrogens onto an oxygen, form something totally harmless to a living thing called water. But we said last time that after breaking the glucose in half, which doesn't require oxygen, then there's a problem if we have oxygen or if we don't have oxygen. If we do have oxygen, these pyruvate sugars are going to enter the mitochondria. And that's what's the mitochondria is represented by what's right below this dashed line. These pyruvate sugars will enter the mitochondria and begin aerobic respiration reactions, reactions requiring oxygen. But last class meeting we addressed the question, what if, for whatever reason, the cells aren't getting adequate amounts of oxygen? They don't have oxygen available. So then, basically, this whole process is going to stop right here. If you don't have oxygen, you cannot break down these sugars, these pyruvate sugars, any further. But you do have a problem of what to do with those hydrogens. The NADs that have picked off those hot potato hydrogens off the sugar, if there's not oxygen available, they cannot transfer them onto oxygen. So we learned that what they do is they retransfer them right back to where they came from, right back onto that pyruvate sugar, And that turns them into either lactic acid, which is what happens in animals, including us, or it's the attaching those hydrogens back onto the pyruvate sugar, it's turned into ethyl alcohol and CO2. That's called alcoholic fermentation, and that's what occurs in plants and fungi, including yeast. So we have to finish just describing alcoholic fermentation, that's where we finished off last time. And then we're going to move on to aerobic respiration. So, in terms of alcoholic fermentation, on page H4, so on H4, we had talked about, on H3 and H4, lactic acid fermentation last time. And we had spoken of what is the limiting factor of why some people run 2 blocks and are exhausted, and other people can run 2 miles before they're exhausted. What did we say is the limiting factor? The condition of your heart and the ability of your heart to pump oxygenated blood rapidly enough to your active muscles so they don't develop lactic acid. So we talked about that up here. Now remember, I hate to keep sounding like a broken record or something. You don't want to take biology again. You want to finish it this semester, right? Unless there's really people who want to have me again. You're enjoying me a lot. So even if you are, you don't want this again. So there's only 5 weeks left. That's it. The next 5 weeks are going to depend, for some of you, are going to determine whether you see me again or another biology teacher. You want to make these 5 weeks count. That's the lesson I'm trying to tell you. Get serious. Whether you may have been serious, get more serious. All right? You don't want to repeat this again. All right, so anyhow, we talked about lactic acid fermentation in plants and in fungi, including yeast. When they are breaking down sugars to make ATP, and there's not oxygen available, the hydrogens are transferred right back onto the pyruvate sugar, but in this case, it's turned into ethyl alcohol or ethanol and carbon dioxide. And so it's because plants and fungi form alcohol and CO2 that we as humans, at least those humans who like drinking alcoholic drinks, have taken advantage of this to use these kinds of organisms, mostly yeast, and add these yeast to various foods and place them in an environment without oxygen so that we can purposely use those yeast to convert the sugars in those foods into alcohol and CO2, creating alcoholic drinks. So, so we started to talk about this at the end of class last time. So a drink that's about as universal as any, now there are places in the world where people don't drink alcohol, and that includes much of the world where Islam is practiced, and also where Mormons live. They don't drink alcohol if they're Orthodox and serious. But anyhow, for most of the rest of the world, most people do drink alcoholic drinks, and an almost universal drink is beer. And one of my goals is to travel the world and taste beer from every single place where it's made, because it's found everywhere. You get beer in China, in the Philippines, in India, in Latin America, everywhere in Africa, everywhere in the world there's beer. Japanese have wonderful beers. So it's a major goal in my life. So we said that you can take these yeast or yeasty beasties and throw them into anything containing sugars, and that includes starch, because starch is just a bunch of sugars. And in an environment without air, they will simply turn those sugars first into pyruvate sugar. Why are they doing it? To make ATP. And then, because there's no oxygen, they convert that pyruvate sugar into alcohol and CO2. So in fact, the sugar that's, the carbohydrate that's used for beer is barley, which has maltose, a grain sugar we learned on page D1. So you throw the yeast in, and in an anaerobic environment, they convert that maltose into alcohol and CO2, and this is called beer. And we said that beer is about 6 proof. We can convert proof into percent alcohol by dividing it by 2, so it's about 3% alcohol. Now, grapes. So there's sugars in grapes. So you mash up the grapes. You throw some yeast in with the grape juice. You put it in a big container where there's no oxygen. And in this anaerobic environment, they will turn the sugars into alcohol and CO2, and that's called wine. Now, most wine that's commonly sold is 24 proof. So if you divide 2 into the proof, that's 2 into 24, or 12% alcohol. Now, that's wine. Now, there's, of course, if the wine not only has the alcohol, but also the carbon dioxide, and there is incidentally carbon dioxide in beer, you see all the little bubbles of CO2. If the wine has carbon dioxide, we call it a sparkling wine or champagne, or asti spumante. That's where it's got the carbon dioxide in it. Sometimes they'll release the carbon dioxide just to have plain wine. And so just plain wine doesn't have the little bubbles of CO2. That's the only difference between sparkling wine and regular wine. As far as why is there so many different kinds of wine, red wine and pink wines and white wines, the answer is because there's lots of different kinds of grapes. Right? So you go to the market, there's green grapes and red grapes and purple grapes, right? And all different kinds of grapes, so they use different grapes and they get slightly different flavors of wine. So, as far as, which wine should you use for which food, who cares? I'm just drinking the wine for the alcohol. But anyhow, so that's why. Now, the problem is that alcohol actually damages and can kill cells. You may have heard that drinking excessive amounts of alcohol can damage brain cells. What did I just say? Okay, sorry. So it can damage brain cells. It can damage liver cells. And in fact, it can even kill the yeast that are turning that sugar into alcohol. So in fact, when you've got these yeast in this big vat, this keg, in its anaerobic environment, turning the sugars into alcohol in order to make ATP is why they're doing it. Actually, when the alcohol reaches about 24 proof or 12% alcohol, it starts killing the yeast. So for that reason, you can't make any alcoholic drink by natural fermentation any more alcoholic than approximately 12% alcohol or 24 proof. So if you want something with a higher alcohol content, and people in the various places of the world have desired something with even more alcohol, then you have to do something called distill. Distill it. Distill it means concentrating that alcohol. What they do is they heat the alcoholic drink up, in this case heating up the wine, and they basically cause the water to evaporate. Because there's water, right? They began by taking the grapes, right, and put some water with the grape juice, and so they're going to heat it up so that the water evaporates, and that concentrates the alcohol, right? And that's called distilling it. That's all distilling it is warming it up and evaporating the water. When you take wine made from grapes and you concentrate the alcohol by warming it up, that's called distilling, you create something called brandy. So brandy is simply distilled wine. And the normal alcohol content of most brandies is about 60 proof. Divided by 2, that's 30% alcohol. Okay, now that's worth drinking. So some of you have heard about all this stuff in lab as well. And it's not so much that I do want you to know about beer and wine. I don't care so much that you know about all these other drinks. But it's kind of fun to talk about. So people have taken all kinds of different foods that contain carbohydrates in them. And they have added the yeast, and in an anaerobic environment, an environment without oxygen, they ferment it and then concentrate the alcohol by distilling it to create various types of stronger alcoholic drinks. So classically, if you took wheat and you fermented it and then concentrated that alcohol by distilling it, that's how you get Scotch whiskey. All right? If you took corn and you fermented it and then concentrated the alcohol by distilling it, that's called bourbon. Like Jack Daniels is a brand of bourbon. If you took potatoes and you fermented it with the yeast in an anaerobic environment and then concentrated the alcohol, that's how you get classic vodka. Vodka, they call vodka. Okay? If you took the cactus, agave, which is indigenous to Mexico and Central America, and you fermented it, right? And it's got carbs in the leaves, sugars, and distilled it, concentrated, you get tequila. If you took rice, which certainly has carbs in it, and distilled it, you get sake, which they obviously make in Japan. If you take sugar cane, which obviously has sugar in it, and you ferment it and then distill it, concentrating it, you get rum, like from Jamaica. So, and some of these, like rum, are typically 100 proof, 50% alcohol. So I could go on and on. You can take apricots or plums. If you take plums and you ferment it and distill it, you call it slivovitz. You can have apricots or peaches, and you get apricot or peach brandy. So anything with carbs in it can be basically fermented and distilled, and it creates all these various types of alcoholic drinks.
Can I ask you a question?
Yeah.
Agave, is that
It's a cactus.
comes from the cactus? So that's the agave that we're using as sweetener? That you can use as sweetener now?
I don't
Yeah,
know.
that's true.
Yeah, because I've been wondering what agave was.
Well, it's a cactus. Yeah.
Yeah.
Okay, I don't know about the use of agave as a sweetener. I only know it for tequila. But you know, I have different priorities.
Well, I understand that, but I was asking.
That's
They're
okay.
using it as a sweetener.
All right, so now that we've talked about alcoholic fermentation, so now we understand what happens when living things are breaking down foods to make ATP, but in an environment where there is no oxygen. All right? Now, most of the time there is oxygen. So now we're going to talk about the aerobic respiration reactions that occur in the mitochondria. Now we wrote that evolutionary biologists believe that this series of biochemical reactions that use oxygen only evolved later after there was already oxygen in the atmosphere. We had pointed out that the original ancient atmosphere of the earth is believed to have had no oxygen. That the source of oxygen came from photosynthesis carried on by plants. And before there were plants, there was no free oxygen in the atmosphere. So these evolved later. In the aerobic respiration reactions, which occur in the mitochondria, only when there's free oxygen available, there will be complete breakdown of the pyruvate sugar molecules, which were formed in glycolysis. They will be broken down completely into carbon dioxide. So we're going to totally break apart the sugars into carbon dioxide. Now, on page H5, we commonly divide the aerobic respiration reactions into three parts. The so-called transition reaction, the Krebs citric acid cycle series of reactions, and letter F, the electron transport system series of reactions. Now, obviously, I've got stuff written here. But before we look at that, and we will, let's look at our diagram on page H10 so we can kind of track this whole big process. So on page H10, where did we leave off? Here's where we left off. Glucose was split in half into two pyruvate sugars. These represent the two pyruvate sugars right here. They're each 3 carbon atoms big. Now, and in addition, 2 NADs removed, each one removed a pair of hot potato hydrogens. Now let's assume oxygen is available. Previously we had assumed there was no oxygen, so lactic acid or alcohol was formed. Now let's assume oxygen is available. The pyruvate sugars enter the mitochondria. That's where these reactions down here are going to be made. And the 3-carbon pyruvate sugar is turned into a 2-carbon acetyl sugar. And we're thinking, a lot of us are thinking, what do you mean? So here's what I drew to try to represent this. Here's the 3-carbon pyruvate sugar. We have 2 of them. I drew arrows, and they have been now turned into acetyl sugars. Each acetyl sugar is 2 carbon atoms big. Can everybody see how I show 2 sugars, each 3 carbons big, have been turned into 2 sugars, each 2 carbons big? These are called acetyl sugars. Now, if we've gone from 3 carbon sugars to 2 carbon sugars, we're missing 2 carbon atoms, aren't we? What happened to them? They're right here. So in other words, when the two pyruvate sugars are turned into two acetyl sugars, we get two carbon dioxides. Those are the two missing carbon atoms. In this process of aerobic respiration, we said we were going to break apart the sugars, the pyruvate sugars, completely into carbon dioxide. So that's what's happening. Now, in addition, additional hydrogen atoms are removed from these sugars as they're being converted from pyruvate to acetyl sugars. And those hydrogens are picked up by the NAD. Now we know there's oxygen available, otherwise this reaction wouldn't even be occurring. This reaction we've called the transition reaction. Now, there is a coenzyme that plays a role, and this is called coenzyme A, CoA. If you went back to section F, where we talked about on page F5, we mentioned the B vitamins, which are involved in cellular respiration. We identified 3 B vitamins on page F5, niacin, riboflavin, and pantothenic acid. And we actually wrote that pantothenic acid, which is a B vitamin, whether you've ever heard of it or not, is actually what makes up this thing called coenzyme A. Just as niacin makes up the coenzyme NAD, and riboflavin makes up the B vitamin FAD, which we're going to talk about in a moment. All right, so that's the transition reaction. Let's summarize it by looking back on page H5. On H5, at the top, so what happened in the transition reaction? The 2 pyruvate sugars were turned into 2 acetyl sugars, plus 2 carbon dioxides were formed, and 2 NADs each picked up a pair of hot potato hydrogens. All right, that's a summary of what happened in that transition reaction. All right, so we went from 2 3-carbon sugars to 2 carbon sugars and 2 carbon dioxides. Now, the next thing that happens is called the Krebs citric acid cycle series of reactions. Before we look at this, let's just show you back on H10. So on H10, what happens next is written right here. It's called the Krebs citric acid cycle. It's known as the Krebs cycle because it was named after Dr. Hans Krebs, who first described it. He was knighted by the Queen, so he became Sir Hans Krebs, and he also won the Nobel Prize for it. Anyhow, and it's known as the citric acid cycle because it utilizes citric acid. Yeah, the same stuff that's in orange juice. All right? Now, basically what's going to happen in this citric acid, Krebs citric acid cycle, is that the 2 acetyl sugars, and here's where I represented them right there, they are going to be broken apart. Each of these 2 acetyl sugars is going to be split apart. We're going to split this apart, forming 2 carbon dioxides. We're going to split this apart, forming 2 carbon dioxides. So that's going to give us a total of, what's written right here, 4 carbon dioxides. So at the end of the Krebs citric acid cycle, the sugars have been completely broken apart into carbon dioxide. You'll notice that the pyruvate sugars were turned into acetyl sugar and 2 carbon dioxide in the transition reaction, and then in the Krebs cycle, the acetyl sugars were turned into 4 carbon dioxide. That's a total of how many carbon dioxides? 6. And so the 6-carbon glucose has been broken apart into 6 individual carbon dioxides. The sugar is gone. It's been totally broken apart. Now, in addition, a whole bunch more hydrogens were picked up. A whole bunch more hydrogens were removed from those sugars. After all, sugars contain hydrogens, carbon, hydrogen, and oxygen. So those hydrogens have been picked up, mostly, again, by the coenzyme NAD, which in reality is the B vitamin niacin. 6 NADs, or 6 niacins, each picked up a pair of hot potato hydrogens. In addition, there's another coenzyme called FAD. It technically stands for flavonoadenine dinucleotide. You don't need to know that. But it contains the B vitamin riboflavin. I'm sure all of you have heard of niacin and riboflavin. Those are B vitamins. They are both involved in transferring hydrogen atoms. transferring hydrogens from the sugar ultimately to oxygen to form water. Now, there's one more thing happened in the Krebs citric acid cycle. The breaking apart of these sugars released some energy, enough energy to phosphorylate ADP into ATP and produce 2 more ATPs. Let's summarize what happened in the Krebs citric acid cycle by returning back to H5. So on H5, in the Krebs citric acid cycle series of reactions, the 2 acetyl sugars were broken apart into 4 carbon dioxides. 6 NADs picked up hot potato hydrogens. That's the B vitamin niacin. 2 FADs also picked up hot potato hydrogens. That's the B vitamin riboflavin. And 2 more ATPs were produced. 2 more ATPs were produced. That's a summary of the Krebs cycle. Now, there's only one last series of reactions. And all of us are thinking, thank God. All right? Only one last series of reactions. The last series of reactions is letter F, the electron transport system. Before we tell you what happens there, let's summarize what's happened so far. So far, in cellular respiration, number 1, glucose, C6H12O6, has been broken apart into 6 carbon dioxides. Number 2, a total of 10 NADs have picked up, each one has picked up a pair of hot potato hydrogens. If in case you're thinking, how do you account for 10? I'll show you in just a moment. In addition, 2 FADs, which are the B vitamin riboflavin, have also picked up hot potato hydrogens. And thirdly, there's been a net gain of 4 ATPs. Let's show you that. Let's show you where that happened. All right? I'll get to this part in just a moment. I'm just focusing on what's happened so far. Everybody got that? So just to show you how we accounted for all that, on this big chart on H10, in our big chart on H10, we have gone from glucose, made up of 6 carbon atoms, into 6 carbon dioxides. All right? The sugar's been broken apart. In addition, a total of 10 NADs have picked up hydrogens. So some of us are thinking, how do you get 10? 2 NADs picked up hydrogens in glycolysis. 2 more NADs picked up hydrogens in the transition reaction. 6 more NADs picked up hydrogens in the Krebs citric acid cycle. What's 2 plus 2 plus 6? 10. And that's what's written right here. Right here, 10. In addition, 2 FADs picked up hydrogens in the Krebs cycle. So right here we've got a total of 10 NADH2s and 2 FADH2s. Finally, so far, we have a net gain of 4 ATPs. 2 ATPs were produced in glycolysis. 2 more ATPs were produced in the Krebs cycle. That's so far 4. All right? So back on page H5, I've shown you how we account for what I've just told you, what has happened so far. So, what needs to happen? What is the last things that need to happen in the last series of reactions? Basically, there's just 2 things that have to happen. Number 1, these hot potato hydrogens that were picked up by NAD, the niacin, and FAD, the riboflavin, have to be transferred to oxygen to form water. We have to show you that. And finally, number 2, we need 34 more ATPs have to be produced because we promised 38, and so far only 4 have been produced. And we promised 38. We've got 4 so far. 34 need to be produced, the majority. So in the last series of reactions is where the majority of ATP is generated. Okay, now the electron transport system is the name of the last series of reactions. We wrote that it consists of a series of coenzymes, both vitamins and minerals, that transfer these hot potato hydrogens from one coenzyme to another and finally to oxygen to form water. Now before I read this and explain it, let's go again back to page H10. On H10, on the lower right, this is labeled on the lower right, the electron transport chain or system. So this is the last series of reactions shown right here. Now, in this last series of reactions, and this is all in the mitochondria, there is a row, a series of coenzymes lined up in a row. These are a number of vitamins and minerals. That's what coenzymes are. One of these coenzymes, one of these vitamins, some of you may have heard of. It's one of the most expensive vitamins they sell in the drugstore. Coenzyme Q10. Has anyone ever heard of coenzyme Q10? That's one of the coenzymes or vitamins that's in this series. There's also iron. Now this is not intended to be a list of all the vitamins and minerals which are really coenzymes that are used in our body. But if you've ever heard of coenzyme Q10, which they sell in the drugstore or in the market now in the vitamin section, it's very expensive. Anyhow, that's one of these coenzymes. Now, before I tell you what's going to happen, I want to give you the same analogy that I gave in my video if you've already watched it. Let's imagine a game we're going to call Hot Potato. So let's imagine we're in the park, we're in somebody's backyard or whatever, and we've got a barbecue grill. And we've got some potatoes wrapped in aluminum foil, and we're cooking the potatoes on the barbecue grill. All right? Let's imagine we get a group of, 6, 7, 10, 12 people in a row. We're going to line them up in a row. And we'll have a fun game. I'm going to go and take some tongs, and I'm going to take these potatoes off the barbecue grill, toss it up in the air. And here's our game. The game is each person's supposed to catch the potato and toss it to the next person in line. So the first person catches the potato as it comes right off the barbecue grill, and it's hot! It's been pulled off the barbecue grill. Tosses it up in the air, the next person in line catches it, it's hot! Tosses it to the third person in line. Now, as this hot potato is getting tossed from one person to the next, what's happening to the potato? It's cooling down. It's giving off heat. It's giving off energy. So at the end of this line, the potato is cooler than it was when it first came off the grill. Right? So now at the end of the line, we've got a cooled down potato that's given off a lot of its energy or heat. So everybody got the analogy of this idea. So what's this got to do here? In reality, we don't have a row of people. We have a row of coenzymes. And what's being passed to them are not hot potatoes, but hot potato hydrogen atoms. These are hot potato hydrogen atoms, and I wrote even hot potato hydrogens right here. These are hydrogens that have just been pulled off the sugar molecule. They haven't been pulled off the barbecue grill, but they've been pulled off the sugar molecule. And they're high in energy. We had a total of 10 NADs that are transferring hot potato hydrogens, and 2 FADs transferring hot potato hydrogens. Now, as these hot potato hydrogens are passed from one, not person to the next, but coenzyme to the next, just like that hot potato, they're giving off heat. They're giving off energy. And just like the potato is giving off heat, these hot potato hydrogens are giving off energy, and that energy is being used to attach phosphate onto ADP and form ATP. Now, what do we call it when we attach phosphate onto an ADP to form ATP? It's called phosphorylation. Now, at the end of this line, at the end of this row of coenzymes, we now have the cooled down hydrogens, just like the cooled down potato. And this is where the oxygen comes in, and the cooled down hydrogens are attached. The cooled down hydrogens are attached to the oxygen to form water. So you'll notice that in this last series of reactions, a whole bunch of ATP is being produced as these hot potato hydrogens are being transferred from one coenzyme to the next. And the cooled down hydrogens are finally being attached to oxygen to form water. And that's how we're getting rid of the hydrogens in a form that's not harmful to the living cells. So overall, this process is known as oxidative phosphorylation. Why is it called that? Because ATPs are being formed, and that's called phosphorylation. And we are getting rid of the hydrogens, aren't we? The hydrogens are being disposed of by being attached to oxygen and forming something harmless called water. And what's the term we use for getting rid of hydrogens and electrons? Oxidation. OIL. Oxidation is a loss of hydrogens. That's how we're getting rid of them. So overall, we call this oxidative phosphorylation. So how many, if you run all these hot potato hydrogens through this series of coenzymes, how many ATPs are generated? 34. So if you form 34 there, that gives us a total of 38. Again, the books may say 36 because it varies in different cells. So let's summarize what we just said back on page H5. On H5, so the electron transport chain or system or phosphorylation chain we wrote is a series of coenzymes that transfers hot potato hydrogens from one coenzyme to another and finally transfers those hydrogens onto oxygen to form water. As these hot potato hydrogens are transferred along, energy is released that's used to join ADP and phosphate together to form ATP. This is called phosphorylation. And the final transfer of hydrogens to oxygen to get rid of them and the process form water is called oxidation. So overall, what do we call this process? Oxidative phosphorylation. All right, this has been my intention to try to make this whole complex process as simple to understand as possible. The real details are far more complex. People who go on to become biology majors and biochemistry majors, they go into all this stuff in tremendous detail because of its importance. But at least this is an introduction. Granted, most of you are not science majors and certainly biology majors, but you have to have some introduction to the knowledge base that we have in terms of this fundamental process of life by which living things take the energy in foods and turn them into ATPs. On page H6, H6. On page H6, now let's try to really make sure we understand what difference it makes to a cell, to living things, whether oxygen is available or not. So, I wrote on H6, comparison of anaerobic respiration versus complete cellular respiration. If oxygen is not available to a cell, to a living thing, then they can only do glycolysis. They can't do the aerobic respiration reactions in the mitochondria. What is glycolysis? Lysis means to split apart. Glyco means glucose, to split glucose apart into two pyruvate sugars. What is our net gain of ATP if all we can do is split glucose in half? We have a net gain of 2 ATPs. We saw that in H10. That's not much. On the other hand, if oxygen is available to the cell, is available to the living thing, then they can do what I call complete cellular respiration. What's complete cellular respiration? That means first the cell does glycolysis in the cytoplasm, followed by aerobic respiration in the mitochondria. They do the whole thing. And that means they will break apart glucose, not just part way into pyruvate sugars, but break glucose apart completely into carbon dioxide and water. And what is our total production of ATP if we can break glucose completely apart into carbon dioxides? 38 ATP. So you'll notice that the amount of ATPs that are produced when you have oxygen available to the cell versus when you don't is huge. 2 ATPs when you don't have oxygen, 38 ATPs per glucose when oxygen is available. That's 36 more ATPs when you've got oxygen than when you compared to when you don't. Now, what's the relevance of this? There's actually some very small, simple, single-celled organisms called bacteria. Those of you taking lab have learned about bacteria. And some of these bacteria can live wonderful, fulfilled lives in an anaerobic environment, an environment where there's no oxygen, because they can actually survive on these low levels of ATP, which is what's generated when there's no oxygen available. These are known as anaerobic bacteria. And so there are living things. Small, simple, single-cell bacteria, very simple, very primitive cells that don't have very high needs for energy, and they can actually survive in a world where there's no oxygen. But most living things cannot. And certainly, cells that cannot survive without oxygen because they can't live on these low levels of ATP are human brain cells. Human brain cells have such a high need for ATP that they have to have oxygen to be generating this much higher quantity of ATP to keep these human brain cells alive. So when somebody stops breathing, their brain cells have 4 minutes left to live. 4 minutes, start counting. So this is why human brain cells without oxygen are dead in 4 minutes. So when people have conditions like heart attacks and strokes, and there's a cessation or stoppage of oxygenated blood flow to the brain, that brain is dead in 4 minutes. All right, so then you've got a human vegetable. Okay, you may have some other parts of the body still working, but the brain is stopped. All right, so the human brain has a very high need for ATP. And most other cells also similarly need ATP, a lot of it, but certainly brain cells do. Now, we next wrote metabolic rate and body temperature. Now, we've mentioned, I actually began with this today by mentioning that as you start to exercise, the rate of cellular respiration starts to speed up because you have to produce more and more ATP. But we know that 60% of the energy released from breaking apart sugars into carbon dioxide actually goes off as heat. Only 40% of the energy is used to attach phosphate onto ADP and form ATP. So, the more you exercise and the faster the rate of cellular respiration in order to make ATP, the greater the amount of heat that's produced. This explains why you get hot as you exercise. It's really the increased speed of cellular respiration that's generating heat in every cell of your body, especially your muscle cells. So that heat is being produced within your cells because most of the energy that was in that sugar is going off as heat. That's predicted by the second law of thermodynamics. Now, there are a number of hormones, chemicals that circulate in our bloodstream that affect our metabolic rate, specifically that can affect the rate of cellular respiration. I've given you two. The first one we learned about originally back in section C. It's called thyroxine. Anybody remember about thyroxine? Thyroxine is the hormone produced by our thyroid gland. It speeds up biochemical reactions in our body. Metabolic means biochemical. If you didn't know that metabolic means biochemical, where have you been for 4 months? We learned it on page A3. All right? So, thyroxine also includes that it speeds up cellular respiration. So, the higher your thyroxine hormone levels are, the faster the rate of cellular respiration, and the faster the production of ATP, and also the production of heat. So this means that people who have hyperthyroidism, excessive amounts, have a very rapid, abnormally rapid rate of cellular respiration. They generate a lot of energy, but they also generate a lot of heat. They're very hot. On the other hand, hypothyroidism, which we talked about back in section C, if somebody has a deficiency, not enough of this hormone, the rate of cellular respiration slows down, and their production of energy in the form of
ATP is slower than normal, and the person feels low in energy and cold all the time because they don't produce the normal amount of heat.
All right? We talked about this way back in section C. We're just pointing out again how thyroxine hormone levels affect energy production and heat production. The second hormone that we've all heard of is adrenaline. Adrenaline, also known as epinephrine, was actually mentioned a few pages ago. You might say, yeah, which pages? We actually mentioned it last class meeting on page H8. On page H8, when we were learning about insulin and glucagon on H8, we learned that not only glucagon, but also epinephrine or adrenaline promotes glycolysis. All right? But anyhow, adrenaline, which is secreted from your adrenal gland, speeds up also your metabolic rate, including cellular respiration. So, adrenaline also, just like thyroxine, speeds up production of ATP and heat production. Now, the question is, thyroxine is always being secreted. Your thyroid gland is always secreting thyroxine to maintain your metabolic rate and maintain the rate of cellular respiration and energy production. But the question is, when is adrenaline secreted from your adrenal gland? And as I wrote on the left, it is secreted when you exercise. So in other words, when you start to exercise, you start to secrete adrenaline or epinephrine from your adrenal gland, and that speeds up the rate of cellular respiration and increases energy production and heat production when you exercise. So it's, in other words, just because you start to run, that doesn't automatically make your cellular respiration and your muscles speed up. It's actually caused in part by the adrenaline secreted into your bloodstream that biochemically speeds up this process. Incidentally, have we ever learned how hormones circulating in our bloodstream would affect the activity of some cells but not other cells? They're called receptor sites. That we learned back in section, when we're dealing with cells, section E. So we learned all about receptor sites. So there are cells that have thyroxine receptor sites and adrenaline receptor sites. Not all cells have all receptor sites for all hormones. But these hormones will affect whatever cells have receptor sites for them. Just reminding you of stuff we've learned earlier. All right, now, the last major topic as we wrap up cellular respiration, I've called the catabolism of fats and proteins for energy. Have we ever learned the word catabolism? Yes, back in section F. Page F1, we needed to know it for the last exam. Be sure you know it for this exam. You didn't know it for last exam. We learned about catabolism and anabolism. Actually, we first introduced those terms on page A3. You needed to know it for the first exam. So the catabolism means the breakdown of fats and proteins, right? Catabolism, catabolic, like catastrophic, things break apart. Now, the way that our body and all other living things work, we'll focus on us, is that our body, our cells, prefer to use carbs, sugars, as the food that they break apart to produce ATP. So most of the time, our cells are breaking down mostly carbs, small amounts of fats, and some fats, I should say, and very small amounts of protein. So our cells right now at this moment are actually using carbs, fats, and proteins that they're breaking down to make ATP. But it's in this order: mostly carbs, some fats, and small amounts of protein. Let's see how this works by looking at page H11, another scary-looking diagram. Okay, this is H11. Now, what does it show on the top? It shows proteins, carbs, and fats, lipids. All three of these are broken apart into carbon dioxide and water in order to make ATP. let's first talk about carbs, because we've already covered this. We know that carbohydrates can be stored in our body as glycogen. Where is the glycogen stored? Which parts of our body store sugars as glycogen? Our liver and muscles. That's not new information. We learned it on page D2. Now, we know that the conversion of glucose into glycogen is called glycogenesis, and that's caused by the hormone insulin. We know that it's a reversible process because the glycogen that's stored in our liver can be broken back apart into glucose and released into our bloodstream. That's called glycogenolysis, and that's caused by the hormone glucagon, and also by epinephrine, adrenaline. Now, this glucose can be used by the cells of our body. It's circulating in our bloodstream. These hormones are what controlling the sugar levels in our blood. And we know that this sugar can be taken up by cells, muscle cells, brain cells, any cells, and it can be broken apart in a process called cellular respiration. Now, we divided cellular respiration into two phases, anaerobic and aerobic, and the dividing line is represented right here by this dashed line. See this dashed line? So what's up here is anaerobic. And basically what anaerobic respiration is, we've already learned this back on page H10, is splitting glucose into 2 pyruvate sugars. So if glucose is represented by 6 carbon atoms, we split it apart into 2 pyruvate sugars. There it is, also known as pyruvic acid. What do we call this process of splitting glucose in half into 2 pyruvate sugars? Glycolysis. The lysis of glucose. All right, then we know that once we form pyruvate sugars or pyruvic acid, if there's a lack of oxygen, what is it temporarily converted into? Lactic acid. How do we get rid of the lactic acid that we form? By inhaling additional oxygen to turn it back into pyruvate sugar. That's called an oxygen debt. Additional oxygen that we breathe to get rid of lactic acid. Now, when we do have oxygen, the pyruvate sugar or pyruvic acid can enter the mitochondria and undergo aerobic respiration reactions. The aerobic respiration reactions in the mitochondria are represented as far as whatever is below this dashed line. The first thing that happens is the pyruvate sugars are broken apart into acetyl sugars. That's called the transition reaction. The second thing that happens is the acetyl sugars are broken apart in the Krebs citric acid cycle into what? Into carbon dioxide and hydrogens that are picked up by NADs and FADs. You can see it's just another diagram showing the same thing we covered in H10. Now, these hydrogens are picked up primarily by NADs, forming NADH2s, but to a lesser extent by FADs, forming FADH2s. Right? NAD is niacin, FAD is riboflavin. The last series of reactions is called the electron transport chain, or electron transport system. You'd say, I don't know where you are right now, I'm all lost. Let me help you. You see these things I circled? That's that row of coenzymes. Didn't we say there's a row of coenzymes, just like there's a row of people lined up in our hot potato game? And incidentally, what's the very first coenzyme in that row? Coenzyme Q10. All right? So here's this row of coenzymes. Didn't we say that just like our hot potato game, these hot potato hydrogens are going to be tossed from the NAD and the FAD, and these hot potato hydrogens are going to be passed from one coenzyme to the next. As they are, energy is released, energy that is used to phosphorylate ADP into ATP. ADP into ATP. So here we can see a whole bunch of ATPs being formed. Finally, what happens to the cooled down hydrogens? The cooled down hydrogens are attached to oxygen to form water. This is just another diagram showing the same thing that we showed you on H10. What do we call this last series of reactions as far as what's happening? Oxidative phosphorylation. Phosphorylation is the forming of ATP. Oxidative is getting rid of those hydrogens. All right, so I've just reviewed basically how carbohydrates like sugars, not only can be stored in our body, but how they are broken down. First anaerobically, or by glycolysis, and then aerobically. First involving the transition reaction, then the Krebs citric acid cycle, and finally the electron transport system. Here, you can see them written. Transition reaction, Krebs citric acid cycle, electron transport system. Also known as oxidative phosphorylation. Now, we said that your body, your cells are primarily using carbs as the source of energy. But our body will also use some fats and small amounts of proteins. Let's see how our body uses fats. First off, where are fats? Where are fats, specifically triglycerides? Where are they stored in our body? In our fat cells. Just like glycogen is stored in our liver cells and muscle cells. Now, the triglycerides are first broken apart into fatty acids. This is a reversible process. That's what this arrow, you notice it has the arrow pointing in both directions. Fatty acids can be 3 fatty acids, can be joined to glycerol to form triglycerides. Is that like new information? No. No. We covered this back in section D. And conversely, triglycerides can be broken back apart into fatty acids. Now these fatty acids can then be converted into acetyl sugars. This is also a reversible process. Sugars can be turned into fats, fats can be turned back into sugars. Hadn't we mentioned that if you eat a lot of sugar, you might become fat? So obviously sugars can be turned into fats. And fats can be